All right. My name's Darrh Bullock. I'm one of the specialists from UK, primarily genetics. I'm housed in Lexington, and I'm not going to introduce the rest of my cohorts. We'll get that done at the end. We appreciate everybody coming out. This is our Beef Seed Stock Symposium. Good turnout today, but it'll also, hopefully, the crowd's small enough that you guys won't be intimidated and are willing to ask questions. We want to keep this as informal as we can. So if you have a question as we go, don't hesitate to raise your hand, stop us, and let's get it taken care of, and then we can continue on. All right, does everybody agree to that? And we want to hopefully have built in some time to have some discussions and things like that. So just to tell you a little bit about the overall project or program that we have going on, this is the Kentucky portion of this. We actually are partnering with University of Tennessee on this. Now we could do that this week. Next week it would have been a little bit harder since, well, I hope we're going to have a football game next weekend. You guys, the Tennessee guys will show up. I'm just not sure if our guys are going to show up, but we hope that they're going to, all right? But anyway, so we're partnering with Tennessee, and so you're going to see quite a few of our Tennessee colleagues that are going to be speaking to you today as well. So the Kentucky portion of this, though, is funded through our partnership, the UK's partnership with Kentucky Beef Network, and we get that funding through the Kentucky Ag Development Fund. And so we're very appreciative of those dollars, and that's how we're able to put on these kind of programs. Now you say, well, we paid money, and you did, but you're going to get a nice lunch out of that, and we have considerable speaker cost and things like that. So I hope that at the end of the day, you're going to say, I got $25 worth of value out of the day. That's what our goal is, and maybe a few more. All right, so I'm going to get us started today, and what I'm going to do is kind of just set the stage, all right, because we're probably at some different levels of maybe understanding of genetic principles and things like that. So I'm just going to kind of set the stage. This is the lightest presentation you're going to get, all right? So if I blow over your head, you might be in a little bit of trouble, but I'm pretty sure I'm not going to, okay? So I'm going to keep it at a lighter level, get us all to the same point, and then we're going to progressively, as the day goes on, you're going to get a little more detail, okay? All right, so we're going to go through the history of selection. Or we think we are. Okay, so selection, as you guys know, this is just the practice of deciding which animals are going to produce the next generation. Okay, it's a very simple concept, but we can utilize it to our advantage, right? We can hopefully make change by making the right selection decisions of those animals, both male and female, that are going to produce the next generation. So how this happens, the methods that we have for selection are obvious. One is the bull or the semen that we purchase, right? We're making a decision, a selection decision when we make that purchase. Bull culling, if we're thinking about this from the commercial side of things, in particular, it doesn't happen, right? It really and truly it doesn't. On occasion somebody may, but they bought the bull, they've bred him. If they found out that was a mistake, they had to pull a bunch of calves and everything else. It's getting pretty late then. They would make a change then, but particularly if it's just poor production, by the time they see that, they've already got the next calf crop coming, right? And so we don't see much going on there. And if they do the right things up front, that shouldn't have to happen very often anyway. Cow calling, what's the number one reason we call a cow? Open. She's open, exactly. What's the likelihood of the cause of her being open? Is it genetics or something else? The greatest likelihood is something else, right? It's probably your fault, management. Is likely the culprit. So we can do some with cow calling. We can impact some genetics a little bit, but really not a whole lot when you get right down to it. Heifer purchases and retention, yes, we can have some influence there as well, but really and truly it ties a lot back to if you did the right job buying the right bull in the first place, particularly if we're keeping replacement females. And so most of the change that we make is through bull selection, okay? And so that's where we have the greatest impact. So that's why from an educational standpoint, we spend so much time on the bull side of things. So we'll go through and talk about from the very beginning how selection happened up to kind of the more modern, current aspects of it. And so the first method of selection in livestock happened based on appearance, okay? And so I'm going to go through a series of few examples that include me as kind of the backdrop on this to hopefully explain what's going on a little bit better. So the first one is going to be, let's assume that Putin goes crazy and he sets off this bomb that kills all the males in the world. No applause from the ladies, please. So all the males have been killed, all right? Except for me. I was down in a cave and I survived, all right? So if the ladies that still survived wanted to perpetuate the human race, then I am the selected male to get that done, right? If I'm the only choice, then I'm there, okay? But I'm a competitor, don't mind competition, and so if I have some competition, I'm hopeful that I would still be the selected choice. Don't tell me if I'm not. And so I hope that I would be. But, you know, there's always a chance for somebody else that survived that was in a cave as well. And in this case, it's going to be Blake. He happened to make it. And so since he was considered the sexiest man alive in 2017, I'm going to assume he's going to be the one that would get selected. And so but does it work? All right, so let's look back at cattle. This is what cattle, it's depicted that they looked like back before really selection started taking place. And so what do you see there? Mixed bag, right? A lot of different colors, all kind of shaggy hair. I think everything has horns. So there's some similarities but differences. And so from that base population, by selecting for appearance, we ended up with many breeds. These are the three most popular, but a lot of different breeds came out of that original base population. And so it's pretty obvious that yes, we can select for appearance and we can make changes that way. So in the area of Scotland there, they chose black polled animals as their choice. Other regions, they chose the red, white face, and so forth. And so that's what made what we consider breeds today. That was the origin of those breeds. So what happens though? Is every animal that's black and polled, is it an Angus? Is every red, white-faced one a Hereford? No. What makes an Angus an Angus? It has an Angus sire and an Angus dam. So, and it's black and polled, all right? So there are criteria, but there also is this thing called a pedigree that we keep up with. And so in order to do that though, that's just not something that just happens. Associations had to be formed, all right? We had to have an association that was developed that would keep up with those pedigrees. All right, and so we're all familiar with that, but that happens in all species. Dogs, the American Kennel Club, you know, we have all the different breed associations here that we deal with cattle. So in the U.S., when we started recording data in these associations, goes back to 1846 for Shorthorn, up in the 1880s for Hereford and Angus. So we've been doing it a long time, been keeping up with these pedigrees and records within these breeds. So in the early 20th century, we started realizing that appearance wasn't the only thing. We could also make selection and make changes to animals on performance basis. How fast do they grow? How, you know, all of those different kind of performance characteristics that we were interested in. And so it was kind of no, hang on, I got to go one more before that. So yeah, performance. So back to my illustration involving me. So we're back to the ladies once again are trying to decide on who's going to keep the human race going forward. And we're going to look at body composition. So here's me a few years back, and that's me with my kids at the beach, and so I apologize for that photo. But if you evaluate me on a body composition standpoint, I'm going to self-evaluate myself as a little light muscled, soft around the middle, a little bit waisty, okay? And so that's what we have. But if you can compare me to others, maybe on the waisty side of things, now that guy's got a lot more muscle than me, right? But he's carrying a considerable more condition than me as well, and we can have the other extreme too, right? And so we have three different body types up there. Is that a genetic thing? There's a genetic component, right? There's a genetic component, but there's also a management component. So how did that guy that big? Well, that's what I researched and found out that's what sumo wrestlers eat. And it's a diet that is heavy in fish, and rice and a lot of calories. In order to get that big, yeah, he has genetic potential for being large, but he has to consume a lot of calories to get there. The guy in the middle, I can promise you he's a meat and potatoes guy. What about the guy on the right? Everybody know who that is? Mick Jagger with the Stones, right? So we all know what he grew up on. Drugs, sex, and rock and roll. All right? So different management, different lifestyles contributed to that, as well as some difference in genetics to go along with it. So with cattle, here's a quote by Jay Lush, and I think it does a very nice job of explaining this relationship. And Dr. Lush, he's kind of the father of modern day animal breeding. The idea of heritability concerns whether the differences actually observed between individuals arose because they started life with different genotypes or were exposed to different environmental forces. Every characteristic is both hereditary and environmental, since its expression can be changed by appropriate changes either in its heredity or in the environment under which that genotype develops. Okay? Pretty simple, but it's important to understand. All the traits we deal with have both components, but there are different levels of influence depending on what traits we deal with. And this is kind of how it breaks down. If we just make it very simplistic, in general, reproduction is what we consider lowly heritable, which means that those traits are slow. Through selection, we can change them, but it's very slow. Okay? It takes us a long time to make significant progress there. Production, that's traits like milking ability, growth, those kind of traits, moderately heritable. That means there's a pretty significant genetic component that we can change through selection, but we also have to pay a lot of attention to management there as well to make sure that we're getting what we want. The end product is the one that's the most influenced by genetics, and that's things like marbling score. Frame score is another example of a highly heritable trait. So those traits, we can make really rapid progress if we make selection practices based on those kind of traits, okay? And so the lower the heritability, the bigger role that management plays. So when I said to you before, when you have those open cows, it was probably management. Could be some genetic components, but probably management. So does that mean we just forget about reproduction when we think about selection for cattle? No. Why not? Because of those, which one has the largest economic impact? Reproduction, right? And so that's why we have to really still pay attention to it, still give it some selection. And I'll be honest with you, it's the hardest one for us to deal with in the beef side of things. And that's where I think we've had some failures as scientists. We haven't given you guys great tools to be able to do this with. And I think that we need to do better in the future. All right, so back to our history of selection. When we think about performance, and in order to be able to select on performance, you have to have data. We have to measure them. In order to change them, we have to measure them, okay? And so to really do a comparison on a performance trait, we have to put everybody on the same playing field. You guys have all heard of adjusted weights, 205-day adjusted weaning weight is the classic one, right? Where we have to make sure that all the animals we're comparing, it's based on that they're the same age, that they're the same sex, that they basically have the same age of mother that raised them, because there's differences in milking ability depending on age. So all of those factors have to go in to adjust the data so that we can put it on a comparable basis. Well, here's the nice thing, that we talked about breed associations and the pedigree, keeping that data. Well, when we really starting to look more at performance side of things, then what the breed associations, they took on another role. They took on the role of when you send us in your registration stuff, send us your raw weights and information, birth dates, all that sort of stuff in as well, and we will compute the adjusted values for you. They actually did the computing of those adjusted values, and they decided that a good way to actually report that was through ratios. All right, and those of you that have been on Earth a little bit longer than some of the others, remember when this was what we selected on, was based on ratios, right? And as a reminder, a ratio is pretty simple. It's that adjusted individual measurement of an animal divided by the average adjusted measurement of that contemporary group it was raised within. So a ratio is specific to that contemporary group, okay? So comparing all the animals within that contemporary group. And then, of course, that division fraction is then multiplied by 100. So how does it work in practice? Well, we have Sheldon and Ken Jennings. The ladies decided that they wanted to make sure that whoever was going to keep human race going was smart, right? So they actually set up an aptitude test to kind of test the potential candidates for how smart they were. Well, the first day they put this test on, Ken Jennings and Sheldon showed up to take the test, and I showed up to take the test. All right, so we all three take the same test, same location, same conditions, right? So we are a contemporary group. And when we ratioed out, those guys kicked my butt, all right? And so if you just didn't know anything else and you looked at those ratio values, you'd probably say, Bullock's pretty stupid. He's just not the sharpest knife in the drawer, right? Because that's pretty low ratio. I can remember back when we were dealing with ratios that it was a very common thing to somebody to say, I'm not going to buy a bull that ratios under 100. I don't buy below average bulls, right? Does that sound pretty familiar? Now, my argument is, well, if that's, if you're only buying above ratios or above 100 ratio bulls, you go to a bull sale with a herd that's really a genetically inferior herd, you can buy that 115 bull and he can be pretty bad, right? Same thing, you go to a genetic herd that has a high genetic potential and buy a 95, that might be a pretty good bull, but he's going to get looked over because he's below average. So, I didn't like this. I'm going to tell you, like I told y'all before, I'm a competitor. I didn't like being ratioed that low. So I decided, I went into a little bit of a depression at first, and there's proof. I kind of let myself go. I didn't shave, didn't cut my hair, didn't do anything. I was just tore up. But they decided they would give another test. Now, they changed it because they didn't want to give me an advantage, so they changed the test, but there's another test, another location, and it happened that some other guys showed up. So Nick Nolte showed up after a pretty rough night of partying, and, whoops, come on, caveman. The caveman showed up, okay? So the three of us took the test. And now, when we did a ratio, all of a sudden I'm brilliant, right? I'm now at 116. Did my aptitude go up at all? No, I just was in a more favorable contemporary group, all right, for me to be able to shine, all right? So that was a real problem with ratios. And what you also find out is when you do this is that it doesn't really matter. Now, we can make some assumptions here and say, yeah, Sheldon's smarter than the caveman. But we don't really have a way to compare those two, right? There's no real good way. They were completely tested, completely different and everything. So that was the problem with ratios. You couldn't compare from farm to farm, even on the same farm, if they were raised differently, right? And year to year, so we had some real issues there. So a guy named Charles Henderson at Cornell developed a system in the 70s, early 70s, that actually was a statistical model to take care of this problem. And we call those breeding values. What he developed was a statistical method of giving us breeding values, which basically is to take pedigree information and data and put it through a statistical model to separate out those contemporary group effects or the management differences between animals, so that what we're looking at is genetic merit and making a comparison based on genetic merit. And this is the basis of EPDs and what we call that go through the National Cattle Evaluation, okay? And so in a very simplistic, and I promise you, you're going to see much more complex explanations of this, so enjoy this one, all right? Because you're going to see the real deal with some of our future speakers. So here's my concept of how it works. We have the pedigree, we have the performance. I'm kind of the same way as a mechanic. I really don't care how that motor works, as long as when I turn the key, it turns on, I put it in drive, it goes, all right? And so this is that version. You're going to get the mechanics version later on. So the pedigree and the performance, we stick it into the machine, and what spits out are EPDs. Now, unfortunately, and this is always the case, anytime you hear about statistics, there's going to be something that tags along with what you want. We want genetic merit, or EPDs. What we get is that and some error, right? That's one of the unfortunate realities of the world, is we're not perfect at being able to predict those EPDs, so there is some error that goes along with that. Now, that model was capable at least of taking out the environmental effects. Prior to that, when people were just doing it based off of those ratios and all, for the most part, that is not being done. It's being done on the micro level within a herd or within a contemporary group, but not across them. And so that's what the beauty of this is. It enabled us to get to that level. So how does it work? And here, once again, is a very simplistic version of how it works. So these are the two guys that we're wanting to compare to see which one is smarter, right? We know what the ratios are, but we really don't know for sure which one is smarter. So how can we get to that? Any ideas? It's what we call connectedness. But the fact that I took the test compared to both of them, right? So now we know the difference between me and Sheldon, that he was smarter than me. And we know the difference between me and the caveman. I was a bit smarter than him. And so now we can kind of predict how much smarter maybe Sheldon is than the caveman. All right? So that's what that statistical model is doing, is giving us looking at these relationships. And this can actually happen up through the pedigree as well. Understanding relationships within the pedigree, this can also go up into the pedigree as well to give us these kind of comparisons. And so that's very, very important to being able to compute our EPDs. So an EPD is a prediction of genetics a bull will pass on to his calves when compared to other bulls within the breed. All right? Very simple. The beauty of it is what it does is it uses the bull's actual measurements. It uses all the ancestral or relative data, so parents, grandparents, siblings, cousins, all that data comes in at the appropriate level, and it takes into account the environment to give us our EPDs, our genetic merit. It's not perfect, as I said. We have that error that comes along with it as well. So we need to convince ourselves and our customers that it's a risk management tool. But it's the best tool we have. It is the best tool we have. And so, and trying to use that tool in conjunction with other tools is not a good idea. using it along, our EPDs along with the actual measurements and all. That's what we call double dipping. We don't need to be doing that. So basically how it works is any EPDs we see, of course, as you guys know, it's on the unit of measurement that we deal with. So for this test, that's going to be test points. Whoops. And so Sheldon ended up with an EPD for test score of 25. I had a 15. The caveman had 5. So anytime that we think about EPDs, an EPD doesn't tell us basically how smart each of us are. What's it tell us? What does an EPD tell us? It's in the title, expected progeny difference. Progeny. It tells us about the progeny, not that individual. And so if we look at it, we have to start with a base. And so let's say that Sheldon's kids' average score have about a 95 for an average score. So my kids, because I'm 10 points below, on average, we would expect them to score around 85, okay? And then I was about 20 points higher than the caveman, so we would expect on average his kids to be about 20 points below mine, okay? Same thing in cattle, as you guys know. And so I'm not going to spend a lot of time here. I know you guys know how it works, but for what we call a direct trait, like weaning weight, the difference in the two bulls, if we use them on same cows, same environment, and looked at their calves, we expect the bull on the left's calves to be about 15 pounds on average heavier than the calves produced by the bull on the right. We look at maternal traits as another generation down the line, right? It's how that bull's daughters' progeny perform, okay? So for a trait like milk, which is truly maternal weaning weight, we call it milk out of simplicity, but there's more to it than that. It's actually maternal weaning weight. If we look at the differences there, the bull on the left, his daughters should raise calves that at weaning weigh about 10 more pounds than the calves produced by the bull on the right's daughters. Okay? So just remember that, and so for your customers, maternal traits are only important if they are keeping back heifers, right? If not, then we can focus just on other traits like growth or carcass or whatever is going to pay them the dollars. All right, so how does genomics play into this? We hear a lot about genomics, and you're going to hear more about genomics later, but I'm going to try and give you kind of a simplistic version of how genomics enter into the picture. Well, we have something called an SNP or a SNP, single nucleotide polymorphism, okay? And what that is if you look at the top bull, the bull number 1, as you see going across there, he and bull number 2 are the same at almost all loci. I mean, and that's throughout the entire genome. They're going to be identical to each other for most of it, all right? There's just a small fraction of things that happen here where we had what's called a mutation or a SNP, and that's where we had a change. So if the wild type was CG, we had a change to TA. So what does that do? What did that do to bull number 2? Well, it depends, right? You're going to hear that word a lot today, I promise you. It depends. What does it depend on? Well, it depends on, first of all, was it in a coding region? Is that a region that's actually producing a protein, right? If so, it still may not. It might not make any difference whatsoever, but it may make a difference. A simple change like that is what causes the difference between red and black coat color, okay? And so there can be, if it's outside of the coding region, then it's not going to directly impact anything. Does that mean it's useless? What do we typically call these? Markers. So it's not necessarily the causal mutation. It could have happened close though, so that when we have segregation, it most of the time travels with the gene that we're interested in. Okay? And so that's a simple version of a SNP. So what we can do is what they did was they looked at 50,000 SNPs across the entire genome and wanted to see what the association was with the different traits. And this is a trait like yearling weight, okay? And so they wanted to look at how it impacted yearling weight. And you can see that for the most part, there's a lot of small effects at each SNP. But there's a couple of big ones, right? There on chromosome 6, there's a major effect on yearling weight. Chromosome 11, there was a pretty good one. Chromosome 14 and 20, but most of them are a lot of little effects, right? And so we can use that information now to help us to compute better EPDs. So how it started though, you guys can remember some time back, it hasn't been that long ago, we started with the star system. Everybody remember the gene stars, or some of you guys remember? And that was just looking at a couple, when you look at that and you see all those different genes, that, the fact that we were trying to make decisions on one or two loci, that was just crazy. Then we got into what we called molecular breeding values, okay? And that's where we were looking at more of that total association. And these were meant to be, they were put out by the genomics companies to be a selection tool similar to EPDs. Now the one difference is a breeding value, you have to cut it in half first, okay? And so for these two bulls with the molecular breeding values, we would assume the bull on the top would have about 10 more pounds of weaning weight, or his calves would have about 10 more pounds of weaning weight than the calves produced by this bull on the bottom, all right? That's all good and fine and dandy, and the technology was solid at that time. But we also then, on those same bulls, you could go to a bull sale and see this, right? Those same two bulls that had EPDs. Bull on the top has a 15-pound, the bull on the bottom a 25-pound. But now the EPDs tell us that the bull on the bottom is going to give us calves that weigh about 10 more pounds than the bull on the top. So conflicting information. And I guarantee you, if you guys were doing this at that time, this was not an uncommon occurrence to see for them not to match up. Does it mean one technology was wrong and the other one was right? It doesn't. And as a matter of fact, and I don't know about my colleagues what they think, I think the technologies were probably similar, comparable. But it's just the different methods because remember what we said before? There's error associated with all of these. And so we wouldn't expect to get it right all of the time. But that was a significant problem. We had to get that fixed. I was suspicious that we never would, but through organizations like the Beef Improvement Federation, we really pushed hard to get these two technologies to merge, and we were able to get that done, and that's to the benefit of the industry, I can promise you. So now, what that National Cattle Evaluation Equation looks like is we still have the pedigree, we still have the performance data. Now we bring genomics into that, and this is going to get, again, explained to you guys in a much better way later. We're still bringing out the environment. So we're still producing an EPD, and I hate to tell you, but it still has error, but the error is smaller now. All right, so that's what it's able to do for us. And so that's the goal of all of this, to improve the statistical model or to improve genomics. Our efforts are always to reduce the level of error that we have to deal with. So genomics now, we have this. So if we don't have genomics, this bull was a calving ease direct of 15, just based on his phenotype, or his own phenotype and his pedigree, with an accuracy of 0.15. If we have him genomically tested, it may change his EPD. And hate to tell you, but every time you get one genomically tested, it's not going to make his EPD more favorable. It's going to make it better. There's a difference between better and favorable, right? The EPD is going to be better, but it might go in the direction you don't want it to go. But we are always going to improve the accuracy. All right, so here's kind of why it happens. We have more accurate parentage now, and we have improved relationships. That doesn't mean we get along better with each other. I'll explain what I mean by improved relationships here in a second. So, to explain this, once again, going to bring myself back into this. This is me and my 4 brothers a couple of years ago. Do you? Well, which one's me? Come on, the guy with the hair. The good-looking one with the hair. No, that's my brother. He's a vet in Florida. He's short and fat and bald-headed. Come on. All right. So show you this because I'm going to use an example from my family again. So a number of years back, let me ask this first. Any of you guys had the human genomics test done on yourself? See, nobody will admit that. There you go. Thank you, Kevin. All right, one poor soul out there. All right, so let me tell you how it works. All right, so if you decide that you're going to do it, you send $100 or whatever it is now to a company. All right, they send you a test tube. You spit in the test tube. You send it back to them. You do not send them a list of your family. You don't say, this is my dad, this is my. You don't send them that. You send them your spit in the test tube and your email address. They send you the list of your family. Okay? There's a difference, right? Well, of the ones that's been tested. That's correct. All right. So here's the deal. So I had the test, first of all, my dad had the test done a number of years back because we were trying to trace some genealogy stuff. So he had the test done. I'm a geneticist, so of course I wanted to have the test done on myself, so I had it done. A few years after that, my son decided he wanted that for a birthday present, so that's what he did, and he got tested. So we have 3 generations that got tested, all right? And so I wanted to get to the next slide I'm going to show you, but to get there on the software that they provide you online, this is what showed up first. And I had really not sat down and thought about what I was about to do, all right? And I pulled this sheet up, and I saw that thing say parent-child. And I had a sinking feeling. And so I look to the left, and I see William Frank Bullock, Jr. And that's the guy I called Daddy for all of my life, all right? So that was a good thing. Do you think that happens for everybody that does this? Sometimes that's Uncle Bob that shows up as your father, or your dad's best friend. If you're going to do it, be careful. Now, here's the next thing. What do you think the next thing I did? I had to make sure that my son was who he was supposed to be. And fortunately, he was. All right? And I'm happy to say my daughter has now been tested, and she is mine too. I'm not sure about the mother, but I know I'm the sire. All right. So if you're going to do it, just be confident in your family members, all right? Because I promise you, I have a good friend who found 2 half-siblings that he did not know he had, all right? So this is what I wanted to get to, though. And this is again, trying to, they had this software that you could pull up, and I can see how much relationship I had with my dad and how much relationship my son has with my dad. So here's the deal. I know you guys have all heard these things before. So with your parents, how much DNA do you share with your mother and your father? 50-50, right? How much do you share with your grandparents? What they never told you is that is an estimate. That's not a hard number. The 50-50 is a hard number. The 25 is not. And this is why. This is how, because we have random segregation of the DNA to make the sperm and to make the egg cells, and then when they combine. All right, so what this is showing you is the red or the orange bars are my relationship with my dad. And so you can see I got a full chromosome of DNA for my dad for all 22 autosomal chromosomes, okay? My son is the light blue, and that's his relationship to my dad. And so you can see here on chromosome 12, he got my dad's full chromosome. So for chromosome 12, he got my dad's full chromosome. No relationship to my mom on chromosome 12. Don't feel sorry for her because she kicked my dad's butt on chromosome 4 and 5. So he got none from my dad for those 2 chromosomes, so he got everything from my mom for those 2 chromosomes, okay? And so, and you can see on some of them it splits, different proportions, so that's why you can technically not be related to one of your grandparents. You can technically not be genetically related to one of your full sibs, which normally you're 50% related to, right, on average. A neat study that Steve Miller did a number of years back was he looked at 6 full sib Angus bulls, which we would expect their relationship to be 50%, right? Because of inbreeding, they were actually 59%. And so he looked at each individual pairs, possible pairs, and looked at the relationship between those 2 bulls. And the 2 bulls that were the least related were only 49% related. The 2 bulls that were most related were 65% related, genomically. Okay? And so it's not, and those little differences help us with those relationships, that pedigree part of doing the national cattle evaluation. And so that's why those little small numbers are important to us. And you're going to get a much bigger dose of that later on. So in general, we've got to make our customers think about targets. We've got to get them focused on making their decision, not just going and buying the pretty bull or you know, the bull with the high numbers for this or that. We need to think about targeted selection. And get them pointed in the right direction, because our tools are really good. So if we don't point them in the right direction, we can get them messed up in a hurry. All right? So let's get them pointed in the right direction. Need to focus on the traits that are of economic importance of us. Not because that's a bull you're having a hard time selling and yeah, you know, and he's wanting your advice. Make sure that it has an economic impact to him, and so he can stay in business and come back and buy more bulls from you in the future. EPDs, for the traits that we have or compute them on, are the best tool we have for selection. We need to own that. We need to convince our customers of that, and we need to get them off of those other things. And please, if you don't do anything, stay around to the very last talk today on the marketing and find out what it is that customers are kind of looking at, and you might be a little bit surprised. Genomic information is great. The genomics that we're getting on these bulls to get these genomically enhanced EPDs is great technology. I think it's well worth the return on investment to both you and your customers. Because think about what you benefit. Everybody thinks about it, you know, the cost share program, we're making you guys do it now and all that, and everything thinks that's because it's going to benefit them. It is. They're now able to buy young bulls with higher accuracies, but think about what you get from information. You get to make selection decisions sooner. You can castrate those bulls that need to be castrated at weaning time or prior to weaning, as opposed to carrying them all the way through and develop them and having all that cost and then realize you have a dud of a bull. It's great information. We need to utilize it. Any questions anybody have? If you want more resources, everybody should have their sire selection manual. ebeef.org is a great website to go to with a lot of fact sheets, videos, things like that for you guys to take advantage of.